Welcome back to the deep dive. Today we're really plunging in, not just dipping our toes, but diving headfirst into a world that's, well, it's all around us, actually inside us too, and yet almost totally invisible.
Hmm.
We're talking about microbiology, the incredible science of these tiny powerhouses that quietly, constantly shape our lives, our whole planet, really.
Absolutely fundamental.
So our mission today is to sort of peel back the layers of this unseen reality. We want to explore the basics, the real foundations of these microscopic entities, and show their profound and often kind of surprising impact on everything. Our health, the food we eat, even the air we breathe.
It's staggering when you think about it.
It really is. How can something so incredibly small be so overwhelmingly significant? There's just so much to figure out, even in what you might call the fundamentals.
Yeah.
And this whole deep dive, shedding light on these hidden architects, is presented by Knowledge Flow.
It's remarkable, isn't it? When we talk about microorganisms, we're genuinely discussing this vast, unseen influence that touches every single aspect of existence.
Right.
And understanding these basics, it isn't just for scientists in labs. It's really foundational for anyone who's curious about, well, the fabric of life on Earth.
Totally.
Despite their minuscule size, these organisms are, essentially the bedrock of biology. They're enabling or influencing almost every process you can imagine, from the grand planetary cycles down to the intimate workings of our own bodies.
Wow.
So yeah, we're about to take a journey, starting with just identifying them and moving all the way to understanding their intricate internal structures, their genetics, their whole place in the web of life.
Okay, let's unpack this then. When most people hear microbiology, I think they often just picture germs, right? Or something vaguely dangerous.
Mm-hmm.
But what exactly is microbiology? It's got to be more than just studying things that make us sick.
Oh, absolutely. Microbiology is, it's this really comprehensive branch of biology. It's dedicated to studying all living organisms of microscopic size.
All of them.
Yep. That includes the bacteria we know, but also fungi, algae, protozoa, and then there are those really enigmatic infectious agents, kind of on the borderline of life, viruses.
Ah, right. Viruses.
So it's a field that tries to understand them completely.
So it's not just what they are, but pretty much everything about them, like their whole life story.
Exactly. Precisely. Microbiology is deeply concerned with their form, their structure, how they reproduce, their physiology, how their bodies function, their metabolism, how they get energy, and of course their classification, how we organize them.
Okay.
But it goes beyond just the individual organism. It's also about their fundamental nature, their complex relationships with each other, and crucially, how they interact with other living things, especially their effects on humans, animals, plants.
Then you mentioned their chemical abilities.
Yes, that's one of the most captivating parts, delving into their astonishing abilities to make these profound physical and chemical changes in their environment, and just as importantly, how they react to different physical and chemical things around them. They're like master alchemists of the natural world, really.
Master alchemists. I like that.
And one striking aspect is just how pervasive they are. Microorganisms are literally everywhere. They're profoundly, intrinsically connected with human health and welfare.
Okay.
And they exist in this fascinating duality. Some are undeniably beneficial, while others are harmful. It's this constant dance, cooperation and conflict.
Can you give us some examples of that duality? Because I think that really brings it home, makes it less abstract.
Certainly, yeah. On the beneficial side, just think about your breakfast. Microorganisms are absolutely crucial in making everyday things like yogurt, cheese, even the wine you might have with dinner.
Right, fermentation?
Exactly. And they're indispensable in producing vital medicines. Penicillin, the classic antibiotic.
Yeah.
Interferon, crucial for immune responses. Industrially, they make alcohol, other compounds. And, significantly, they play this critical, often unsung role in processing waste, domestic industrial waste. They're like the planet's tiny, tireless recycling crew.
Wow. Okay. Recycling crew.
But then, on the flip side, we have to acknowledge their negative impact. They are responsible for many diseases that affect us. We'll get deeper into that later, but it's part of the picture.
Sure.
And a key point to remember is that most microorganisms are unicellular, single-celled. And this simple fact dictates so many of their unique traits and behaviors, makes them fundamentally different from, say, us.
So single-celled, mostly invisible. How do we even start to understand them? How do you tell them apart if you can't see them easily?
It must begin with their characteristics,
Yeah.
Knowing what makes them unique. That's
Right.
exactly right. When we meticulously learn the characteristics of an organism, It gives us the data we need to compare with others.
Ah,
Allows us to see the similarities, the differences. That's the absolute first step in identifying and classifying them properly.
okay.
And we use a whole toolkit of characteristics for this. We look at their morphological characteristics, basically what they look like, their shape, size, arrangement.
Right.
And we dive into their genetic characteristics, their DNA blueprint. We look at their ecological characteristics, where and how they live, their chemical composition, their metabolic characteristics, how they process energy and nutrients.
Ah,
Their cultural characteristics tell us how they grow in the lab. Their antigenic characteristics relate to how our immune system might recognize them. And finally, yeah, we even consider their pathogenicity, their ability to cause disease.
mm.
It's a really holistic approach.
Wow, that's a lot of angles.
It is.
Yeah.
And if we connect this to the bigger picture, once we've characterized them, classification becomes the vital next step, bringing some order to this huge invisible world. In microbiology, the taxonomic units, the taxa, they're initially built from strains. Strains are basically all the descendants of a single pure culture. Think of it like a distinct family line.
A lineage.
A lineage, yes. The basic taxonomic group, the fundamental unit, is the species. That's defined as a collection of strains that share very similar characteristics.
Okay.
And within a species, there's often a special strain called the type strain. This is super important because it's the designated permanent reference specimen for that species, like the original blueprint or the gold standard.
Ah, so it's like a scientific fingerprint, making sure everyone's talking about the exact same thing.
Precisely. It anchors the definition. Then, from species, we move up the hierarchy, grouping similar things into broader categories. A family is a group of similar are groups of related species.
Got it.
An order groups similar families. A class groups orders. A division groups classes. And finally, a kingdom groups divisions.
It's like those Russian nesting dolls, but for microbes.
Huh? That's a good way to put it. And taxonomists, the people who do this classification, they strive for two critical qualities: stability and predictability.
Stability. Meaning the classification shouldn't need constant massive changes, even when we learn new stuff. That sounds tough with microbes that change so fast.
It is a challenge, absolutely. But the goal is to create classifications that only need minor tweaks as new info comes in, so the system stays useful over time.
Okay.
Predictability means that if you know about one member of a group, you should be able to infer or predict that other members in that same group likely share similar characteristics. That makes the system powerful for research and communication.
Makes sense.
As for how we classify bacteria, the methods themselves, well, there's numerical taxonomy using math models. There's the more traditional intuitive method based on expert judgment. And increasingly, there's genetic relatedness, looking directly at DNA sequences.
Right. And how do we actually see these things? You said they're microscopic. You can't just, pick one up and look. This is where microscopes are essential, isn't it? Those tools that first opened up this whole invisible world must have been mind-blowing for the first people.
Oh, absolutely. Game-changing.
Yeah.
We primarily rely on two main types of microscopes, and they differ based on the fundamental principle they use for magnification. We have light, or optical, microscopes, and electron microscopes.
Okay, light microscopes first, like the ones in a school lab.
Basically, yes.
Yeah.
Light microscopy uses visible light waves. Magnification comes from a system of carefully ground optical lenses. And within this category, there are several important subtypes, each giving a different view. Well, there's bright field, where the specimen looks dark against a bright background. That's the most common. Dark field makes the specimen appear bright against a dark background, good for seeing thin things. Fluorescence microscopy uses special dyes that glow under certain light. And phase contrast enhances the contrast of transparent specimens, so you can see internal structures better without staining. Each gives unique insights.
And then electron microscopy, that sounds way more powerful, uses electrons instead of light.
Exactly. It is significantly more advanced. Instead of light waves, it uses a focused beam of electrons to create the image. This allows for vastly higher magnification and resolution.
So you can see really tiny details.
Absolutely. We can peer into the minute, intricate details of these organisms, see structures that would be utterly invisible, even with the best light microscopes. These tools were, and still are, fundamental. They let us go from abstract ideas to tangible, observable realities.
Okay, let's dive into bacteria specifically then, because they are just countless. Billions in a pinch of soil, you hear. And they're everywhere. Seafloor, icebergs, boiling hot springs, places nothing else could live. What makes them so incredibly successful, so widespread? What's their secret sauce for dominating these extreme places?
That's a crucial question. And Their huge distribution really speaks to their incredible metabolic adaptations.
Hmm.
They have this unique ability to thrive in environments with unusual chemical conditions, unusual temperatures, places that would be totally hostile to most other life.
Right.
Their biological success largely comes from these metabolic capabilities, combined with their tiny size, less resource needed, right? Plus incredibly fast reproductive rates,
Hmm.
allowing populations to explode quickly. And for many, the ability to build those resistant spores we mentioned.
The little time capsules.
Exactly. Like microscopic time capsules. They let them survive terrible conditions, just lying dormant until things get better. It's also worth noting, not all bacteria have things like flagella for moving or pili for sticking, which we'll get to. But these collective adaptations really explain their ubiquitous presence.
So visually, how do we identify them? Is it mostly about their shape and how they cluster together when you finally get them under the microscope?
Absolutely. The most common, most fundamental method for identifying bacteria often relies on their basic shape and how they arrange themselves in groups. There are three main shapes that cover the vast majority of bacteria.
Okay, shape number one, the simplest.
The first is the coccus shape. These are spherical, basically round cells. Sometimes they look a bit flattened when they're squished together in groups.
Yeah.
And what's fascinating is how these simple round shapes arrange themselves in diverse patterns. And these patterns aren't just random. They tell us something about how they interact. Cocci can occur singly, just one cell. In pairs, called diplococcii. Groups of 4, tetrads. Long chains like beads, those are streptococci.
Like strep throat.
Exactly, like streptococcus that causes strep throat. They can form grape-like clusters to
No,
staphylococci.
staph infection.
Right, staphylococcus. Or even neat cubes of 8 cells called sarcinae. Those streptococci chains, for example, are great at forming biofilms, which is why they can cause persistent infections. The staphylococci clusters help them spread on surfaces.
Wow, okay. So much variety from just a round shape. What's the second basic shape?
The second is the bacillus shape. These are rod-shaped bacteria. Their length can actually vary a bit depending on the environment, making them adaptable.
Uh-huh.
Like cocci, they can be found singly, in pairs, or in chains. This rod shape often gives them a higher surface area to volume ratio, which can be good for absorbing nutrients or getting rid of waste.
Any examples?
Sure. A well-known example are the coliform bacteria. They're used as indicators of wastewater pollution in water testing. Some are also responsible for diseases like typhoid fever. Their presence tells you something important about water safety.
Okay, rods. And the third shape? Sounds like it might move differently.
The third is the spiral shape. These bacteria have a curved form, ranging from just a gentle curve to a full-on, tightly wound corkscrew spiral. Many spirals are rigid and, as you guessed, they're often modal, capable of movement, sometimes with a distinctive drilling motion. A special group within these are the spirochetes. They're particularly slender, long, and flexible. This flexibility lets them navigate through thick, viscous stuff like tissues.
Ah, like Lyme disease.
Exactly. The bacterium causing Lyme disease is a spirochete. Syphilis, too. Their shape helps them bore into tissues.
Amazing how form fits function even at this scale. You mentioned size earlier. What's the typical range? Are they all super tiny or is there some variation?
Oh, there's astonishing variation. It's like the animal kingdom,
Oh.
from a blue whale down to a tiny insect. Bacteria also vary hugely, from about 1 millimeter diameter, which is huge for a bacterium, down to just 20 nanometers in length.
Wow.
But most bacteria we commonly encounter are much smaller, usually around 0.2 micrometers in diameter and maybe 2 to 8 micrometers long.
Okay.
Even the largest known bacteria like Thiomargarita navibiensis or Epilepicium fischelsoni, these giants of the bacterial world, you still need a microscope to see them. Puts microscopic into perspective, doesn't it? So much life operating beyond our sight.
Definitely. Okay, so beyond the basic shape, what's on the outside of a bacterium? Structures outside the cell wall that help it interact, move, defend itself.
Right. These external structures are absolutely critical for survival, movement, interaction with the environment, with hosts. They include the glycocalyx, flagella, axial filaments, and fimbriae and pili.
Glycocalyx first sounds kind of sticky.
It often is. The glycocalyx is this gelatinous covering made of polysaccharides and polypeptides, like a sugary, protein-rich coat. Certain types, called capsules, are really important. They act as a defense mechanism.
So?
Well, they protect pathogenic bacteria from being engulfed by our immune cells, phagocytosis. Makes them slippery, hard for immune cells to grab onto. Capsules also help bacteria stick to surfaces, letting them colonize tissues or form biofilms. They prevent the cell from drying out, and can even be a nutrient reserve.
Multi-purpose coat. And flagella, those are definitely for movement, right? Like
Yeah.
tiny propellers.
Yes, exactly. Flagella are long, whip-like filaments that act as propellers. Each one has three parts: the filament itself, a hook connecting it to the cell, and a basal body embedded in the cell wall and membrane that powers the rotation.
They rotate.
Yes, unlike eukaryotic flagella that whip back and forth, prokaryotic flagella spin like a boat propeller, pushes the cell through liquid. Motile bacteria use them for taxis-directed movement. Positive taxis is moving towards something good like food. Negative taxis is moving away from something bad like a toxin.
Clever.
And the protein making up flagella, the flagellar or H protein, can act as an antigen, something our immune system can recognize.
What about the spiral spirochetes we talked about? Do they use flagella or something else for that corkscrew movement?
They have a special trick: axial filaments. These are found only in spiral cells, especially spirochetes. They enable that characteristic corkscrew motion. Imagine flagella, but instead of sticking out, they wrap around the cell, underneath an outer sheath.
Oh, interesting.
When these filaments rotate, they cause the whole spirochete cell to twist and undulate. This lets it corkscrew its way through viscous stuff like mucus or tissues, vital for their infectivity.
And the last ones, fimbriae and pili. They sound smaller, maybe more for sticking.
They generally are. Fimbriae and pili are small, thin, hair-like appendages, usually shorter and straighter than flagella. Fimbriae are mostly about sticking, adhering to surfaces, like tiny grappling hooks, essential for colonization.
Okay. And pili.
Pili have a more specialized and really significant role. They connect cells for the direct transfer of DNA from one bacterium to another.
Whoa, so that's how they share genes.
Yes, this is a crucial point for understanding bacterial genetics and adaptation. It allows traits like antibiotic resistance to spread rapidly through a population.
Okay, that's huge. We'll definitely come back to that.
Absolutely. So connecting these external structures, they're not just decorations. They are key to how bacteria interact with their world, and especially with hosts. They're the primary interface. Now, should we go inside?
Beneath the cell wall.
Yes. Let's peel back the next layer. What's the first thing inside the wall? The cell's boundary, its skin.
Indeed. The outermost living layer, right under the cell wall, is the cytoplasmic membrane. It's vital. About 7.5 nanometers thick, made mostly of phospholipids and proteins. It acts like the cell's gatekeeper, or a highly selective filter.
It's a hydrophobic barrier, meaning it stops most water-soluble molecules from just wandering in or out, but it actively controls what does get transported across, essential for maintaining the cell's internal balance. Selective permeability.
Okay, the gatekeeper. What happens if you manage to remove that rigid outer cell wall? Does the cell just pop?
Good question. If you remove the wall, you get what's called a protoplast. That's basically the cytoplasmic membrane plus everything inside it. Imagine taking the shell of a hard-boiled egg. The protoplast is the fragile inside part.
How do you make one?
For gram-positive bacteria, you can use an enzyme like lysozyme. It dissolves their main cell wall component, peptidoglycan. Without the rigid wall, they usually become spherical and very fragile.
And for gram-negatives, you mentioned their walls are more complex.
Yes, it's slightly different. Gram-negative bacteria have that outer membrane outside the peptidoglycan layer. So if you treat them and don't completely remove the wall, but the cell loses its shape and becomes round, it's called a spheroplast. It still has some outer membrane bits and maybe residual peptidoglycan. It's like a partially stripped down cell, distinct from a true protoplast.
Do bacteria have complex internal membrane systems, like our cells do with mitochondria and stuff?
Some do, but it's organized differently. Many bacteria, especially gram-positives, have these inward foldings of the cytoplasmic membrane called mesosomes. They look like convoluted tubules and vesicles. People have thought they might be involved in cell division or DNA replication, but their exact function is still kind of debated. Also, in some photosynthetic bacteria, like cyanobacteria, you find distinct intracellular membranes separate from the main cytoplasmic membrane. These usually contain the pigments needed for photosynthesis.
And the jelly-like stuff filling the cell, where all the action happens.
That's the cytoplasm, the granular, viscous material filling the cell, bounded by the cytoplasmic membrane. It's a really bustling internal environment. It's packed with these macromolecular or RNA protein bodies called ribosomes.
Ribosomes, the protein factories.
Exactly. Ribosomes are absolutely crucial. They're the tiny factories that synthesize proteins, translating the genetic code from DNA into the functional enzymes and structural bits the cell needs.
Okay. Now, one of the biggest differences between bacterial cells and ours is how their DNA is organized, right? We have that distinct nucleus with a membrane around it.
This is a fundamental defining difference. prokaryotic bacteria versus eukaryotic cells like ours. Bacterial cells do not have a distinct membrane and closed nucleus, nor do they have that mitotic apparatus like spindle fibers for cell division.
So where is their DNA?
Instead, they have an area usually near the center of the cell that we call the nuclear structure, or often the nucleoid. The cell's entire DNA, its chromosome, is confined to this area, but there's no membrane around it. It's essentially a condensed mass of genetic material just floating in the cytoplasm.
Wow, okay. Big difference. And finally, those spores again. Really important for survival, you said. What's the difference between endospores and exospores? They both sound tough.
They are incredibly tough. Spores are highly significant for bacterial survival. They're dormant, highly resistant forms of the cell, built to endure extreme adversity. Endospores are formed within the vegetative cell, literally inside.
Endo means inside? Makes sense.
Right. They have thick walls, they're highly refractile, meaning they shine brightly under a microscope, a sign of how dense they are. They're produced by specific genera, like Bacillus think anthrax and Clostridium think tetanus, botulism. These endospores let bacteria survive extreme heat, radiation, drying out, disinfectants, sometimes for centuries.
Centuries. Amazing. And exospores.
Exospores, in contrast, are formed external to the cell. The methane-oxidizing genus Methylacinus does this. They form by budding at one end of the cell. Different formation mechanism, but a similar survival purpose.
So these tiny organisms have incredibly sophisticated structures, inside and out, letting them survive and thrive. But how do they multiply so fast? And how do we track that growth? You can't count them one by one.
Excellent question. And it's important to clarify what growth means in microbiology. Unlike, say, a person getting bigger, bacterial growth usually refers to changes in the total population, an increase in the number of cells rather than the size of one individual cell. Growth means an increase in number beyond the original amount you started with, the inoculum.
Okay, population growth.
So this raises the question, what are the main ways bacteria reproduce to cause this often explosive population increase?
Right.
By far the most common and most important mode during the usual growth cycle is transverse binary fission.
Binary fission sounds simple. Elegant in its simplicity. A single cell elongates, then develops the dividing wall, a septum right across the middle, and splits into two genetically identical daughter cells. It's beautifully timed, too. It doesn't start until the cell has fully duplicated its chromosome through DNA replication. The completion of DNA replication actually triggers the division. And new cell wall material is made right there at the septum, and this new material becomes half the wall for each new daughter cell. It's a remarkably efficient multiplication.
So one cell becomes two, two become four, four become eight. Perfect exponential growth, potentially.
Potentially, yes. But are there other ways they multiply? Perhaps less common methods?
Yeah.
Yes, there are alternatives showing their diverse strategies. Some bacteria, like Rhodopseudomonas acidophila, reproduce by budding. A small bulge, a bud, develops on the cell. It grows, becomes a new, smaller cell, and separates from the parent, kind of like yeast.
Another way is fragmentation. You see this in bacteria that grow in long, branching chains or filaments, like Nocardia species. These filaments can simply break into smaller pieces, either rod-shaped or round, and each piece can then grow into a new organism. Different ways to achieve the same goal: more bacteria.
If we put this all together then, how does a bacterial population actually grow over time in, say, a lab culture? What does that typical life cycle curve look like? It can't just be exponential forever, right? That would be terrifying.
No, definitely not unchecked forever. The growth cycle of bacteria in a closed system, like a lab flask, follows a very distinct, predictable pattern. We represent it with a curve that has four characteristic phases. There's the lag phase, the exponential phase or log phase, the stationary phase, and finally the decline phase or death phase. Each phase reflects the population's interaction with its limited environment.
Okay, walk us through them. Lag phase first often labeled A on the curve. Is it just them getting ready, a warm-up?
That's a good way to think of it, yeah. During the lag phase, the individual cells might actually increase a bit in size. They're very active physiologically, making new protoplasm, essential enzymes, metabolizing nutrients, basically gearing up for rapid growth.
But not dividing yet.
Right. Despite all this internal activity, there's a distinct lag in cell division. They're adapting to the new environment, accumulating resources. Cell division really only kicks off at the end of this phase, once they're fully prepared.
Then comes the exponential phase, phase B. It sounds like that's where the population explosion happens.
Exactly. During the exponential, or log phase, the cells divide steadily at a constant maximum rate for those conditions. If you plot the logarithm of the cell number against time, you get a perfectly straight line that shows the consistent doubling. Cells in this phase are considered the most uniform, physiologically speaking. That's why researchers often use log phase cultures for experiments on metabolism. Their predictable growth allows for precise measurements, their peak performance.
But they can't keep that up forever in a closed flask. Resources must run out. What makes them slow down and hit the stationary phase, see?
You're absolutely right. Endless exponential growth is impossible with finite resources. The growth stops or slows dramatically in the stationary phase for a couple of main reasons. Primarily, it's because they've used up some essential nutrient in the medium. They've eaten all the food, basically.
Okay.
Less often, it might be due to the buildup of toxic waste products from their own metabolism. They pollute their own environment. During this phase, the total population number stays relatively constant for a while. Maybe division stops completely, or maybe the rate of new cell formation is balanced by the rate of cell death. It's a plateau.
And eventually things go downhill. The decline phase,
Yeah.
the crash after the boom.
Correct. During the decline, or death phase, the number of viable cells starts to decrease, often exponentially. It's basically the reverse of the log phase. The environment has just become too hostile, nutrients gone, maybe too toxic.
Do they all die at once?
No, it's important to note, bacteria die at different rates, depending on the species and the exact conditions. Some die off quickly, others might hang on for longer. But the overall trend is downwards.
Okay, that growth curve makes sense. Now, how do these tiny organisms actually do all those incredible chemical changes we talked about earlier? Breaking down waste, making yogurt. It sounds like serious chemical heavy lifting for something so small. How do they manage it?
It is serious heavy lifting, and the absolutely essential players, the workhorses, are enzymes. Enzymes are these remarkable substances present in every living cell, capable of speeding up the thousands of chemical reactions needed for life.
No life without enzymes.
Pretty much. Any significant problem with enzyme activity leads to profound changes in the cell, or ultimately its death. You really can't have life without them. And they don't work alone. They function in precise sequences, these reaction pathways, like tiny, efficient assembly lines within the cell. One of the most amazing things is how enzymes act as catalysts. They speed up reactions by incredible factors, millions, even billions of times, without being consumed in the process themselves.
But they're different from, say, a platinum catalyst in chemistry.
Very different. Unlike inorganic catalysts, enzymes are organic compounds, specifically proteins, produced by living organisms. So we can define an enzyme as an organic catalyst produced by a living cell. Now, every enzyme is made inside the cell, but some, called exoenzymes, are secreted outside the cell.
Why would they do that?
To break down large molecules in the environment, like big proteins or starches, into smaller pieces the cell can then absorb. Others, endoenzymes, work entirely inside the cell on internal processes. But regardless of where they work, their general characteristics, specificity, efficiency, sensitivity are the same.
So how do they actually work their magic, that enzyme-substrate complex thing? What's happening there?
Right. The basic reaction looks simple on paper. Enzyme E plus substrate S, that's the molecule the enzyme acts on, combine temporarily to form an enzyme-substrate complex.
Oh.
This complex is transient, only lasts a moment. During that time, the enzyme facilitates the chemical change in the substrate. Then the complex breaks down, releasing the product P and regenerating the original enzyme, E, which is now free to grab another substrate molecule and do it all again.
And the key is lowering energy barriers.
Exactly. The main function is to lower the activation energy needed for the reaction to happen. Think of activation energy as a hill the reaction needs to climb over to get started. An enzyme is like digging a tunnel through that hill. It makes the reaction happen much, much faster with less energy input than it would otherwise.
A tunnel through the energy hill. I like that analogy.
So this raises the question. With so many different reactions happening, how do we categorize these amazing catalysts? To bring some order, enzymes are systematically divided into major classes based on the type of reaction they catalyze, like classifying tools in a workshop based on their job.
Okay, let's run through the main classes.
Sure. First, oxidoreductases. They handle electron transfer reactions. Think of them as energy movers, shifting electrons around, vital for things like respiration.
Got it.
Then transferases. They transfer functional groups from one molecule to another. They're the group shifters, essential for building complex molecules.
Okay.
Hydrolases. They perform hydrolysis, breaking chemical bonds by adding water. These are the molecular scissors, often breaking down large food molecules, like ANSIs. They add groups to or remove groups from double bonds, but without hydrolysis, kind of adders or removers of specific groups.
Right.
Isomerases. They catalyze isomerization, rearranging atoms within the single molecule to form an isomer, basically reshaping molecules.
Okay.
And finally, ligases. These form new chemical bonds, and this process usually requires energy, typically by breaking down ATP. Ligases are like the cell's construction workers, building new molecules, and ATP is their energy currency.
ATP, the energy currency.
Right.
So these enzymes are highly specific tools, but are they always working at full speed? Or can things like temperature or acidity affect how well they work?
Oh, absolutely. Their activity is definitely not constant. They're very sensitive to conditions. Several key factors influence enzyme activity significantly. Temperature is critical. Enzymes have an optimal temperature range. Too cold, they slow down. Too hot, they can denature, permanently lose their shape, and stop working.
Like cooking an egg white.
Exactly like that.
Oh.
Then there's the concentration of substrate. More substrate generally means a faster reaction, but only up to a point where the enzyme gets saturated, all its active sites are busy.
Makes sense.
The concentration of the enzyme itself matters too. More enzyme, faster reaction, assuming there's enough substrate. And finally, pH, the acidity or alkalinity. Enzymes have very specific optimal pH levels. Move too far from that sweet spot and their activity drops dramatically, or they might even denature. Maintaining stable internal conditions is vital for keeping enzymes happy. And connecting this back, understanding how to control and prepare enzymes is crucial, not just for studying them in the lab, but for using them in industry medicine, food, detergents, you name it. We need ways to isolate and work with them.
How do microbiologists do that? Prepare enzymes for experiments?
There are three main techniques. First, growing culture techniques. You basically grow the bacteria in a broth, then put them in a medium with a substrate you're interested in. Then you watch to see if the substrate disappears and the product appears, observing the enzyme working in a living system.
Okay. What if the cells aren't actively growing?
That leads to resting cell techniques. You grow the bacteria first, then harvest them. These resting cells aren't dividing much, but they're still alive and have their enzymes. You add these cells and the substrate to a testing unit and measure the reaction. It's a bit more controlled.
And if you want just the enzyme, no cells involved.
That's cell-free enzyme techniques. You prepare a concentrated suspension of resting cells, then break them open and extract and purify the enzymes, getting rid of all the other cell bits. Then you test the activity of these isolated enzymes with the substrate. This gives the most precise look at the enzyme's function itself.
Do cells just make all their enzymes all the time? Or can they switch production on and off depending on what they need? That seems smarter, energy-wise.
That's a great observation. It speaks volumes about microbial efficiency. Enzymes are broadly split into two groups based on how their synthesis is regulated. First, constitutive enzymes,
Constitutive.
meaning they're always generated by the cell, usually found in fairly constant concentrations, regardless of whether their substrate is around. Think of them as the housekeeping enzymes needed for basic, everyday functions like glycolysis, breaking down common sugars. They're like the lights that are always on.
Okay, always ready. But then there must be ones that only appear when needed.
Exactly. Those are inducible enzymes. They're made only when their specific substrate is present. The cell produces them only when required. It's a brilliant way to save energy and resources.
How does that work?
The process is called enzyme induction, and the substrate itself acts as the inducer, signaling the cell to start making the enzyme. For example, a bacteria might only make the enzyme to break down lactose if lactose is actually in its environment. Lactose gone, enzyme production can switch off.
Super efficient.
This leads to the important question of how cells precisely regulate all these complex metabolic pathways, making what they need, when they need it, no waste. They have sophisticated internal control systems. One is energy link control.
How does that work?
Here, certain molecules, often related to the cell's energy status like ATP, the energy currency, or other related nucleotides, directly influence enzyme activity. They act as signals, telling an enzyme if the cell has plenty of energy or building blocks, and thus whether to speed up or slow down a particular pathway.
So if the cell's loaded with energy, it might slow down the energy-making pathways.
Precisely. Incredibly efficient. Another key mechanism is feedback inhibition. This is a classic regulatory strategy. It prevents the cell from making too much of something, like an amino acid or a nucleotide.
How?
The end product of the metabolic pathway actually inhibits an enzyme that functions early in that same pathway. It's like the finished product sends a message back to the start of the assembly line saying, okay, we have enough, stop making more. Prevents wasteful overproduction.
Neat. And what about precursor activation? Sounds like the opposite, turning things on.
Kind of, yes. Precursor activation happens when an intermediate molecule that appears earlier in a pathway actually stimulates the activity of an enzyme later in the sequence, often the last one. This helps pull materials through the pathway efficiently, ensuring that as building blocks become available, the final steps speed up to use them. It prevents bottlenecks. All these control systems, especially how they couple catalytic activity to the cell's needs, allow for incredible metabolic precision and economy.
Okay, so enzymes are the tools, the gears. But what's the big picture of how microbes get and use energy? This brings us to microbial metabolism, right? The whole chemical symphony.
Exactly. Microbial metabolism covers every chemical activity a cell performs.
Hmm.
Basically boils down to two general types of processes: energy production, how the cell gets its fuel,
Hmm.
and energy utilization, how it spends that fuel.
And what do they spend it on? Seems like a lot for a tiny cell.
It is a huge energy budget. Energy is used for a vast array of things: constructing cell parts, like the wall, synthesizing enzymes and other chemicals, repairing damage, general maintenance, just keeping things running, growth, obviously, actively collecting nutrients from the environment, sometimes concentrating them inside, pumping unwanted stuff out, and motility, moving around, all require energy.
Wow, how do they produce all that energy? Do they always need oxygen like we generally do? Or do they have other tricks?
This is where microbial adaptability truly shines. The diversity of energy production methods is remarkable. They use both anaerobic processes, which happen without oxygen, letting them live in oxygen-deprived places, and aerobic processes, which do require oxygen, like ours.
What are some anaerobic ways?
Anaerobic pathways include glycolysis, that fundamental sugar breakdown pathway, also the pentose phosphate pathway, the Ender-Dudorov pathway, and various types of fermentation, producing things like alcohol or lactic acid as end products.
Anaerobic, like us.
Aerobic processes, which usually yield much more energy, include the tricarboxylic acid cycle, the Krebs cycle, the glyoxylate cycle, also the breakdown or catabolism of lipids, fats, and proteins for energy. Some can even do heterotrophic CO2 fixation, incorporating carbon dioxide in unique ways. This metabolic flexibility is why they can live almost anywhere.
Okay, so they make the energy. How is it used then? What are the main categories of energy spending?
Once the energy is generated, it's carefully spent through two main categories: biosynthetic processes and non-biosynthetic processes.
Biosynthesis, making stuff.
Right. Building things. Synthesis of organic cell material, like in chemoautotrophs, that build everything from inorganic sources. Synthesis of small molecules, like amino acids, the building blocks. And synthesis of macromolecules, proteins, DNA, RNA, the core machinery, and genetic code.
And non-biosynthetic, things other than building.
Yes. This includes crucial activities like the active transport of nutrients, spending energy to pump needed stuff into the cell against a concentration gradient. And, as we mentioned, bacterial motility, energy spent spinning flagella or moving in other ways. Every aspect of a microbe's life needs this carefully managed energy flow.
Okay, we've covered what they do, how they work internally, but how do they pass traits on? And why are they so darn adaptable? Always changing, resisting antibiotics. This gets us into bacterial genetics, right?
Absolutely. Genetics is the fundamental study of inheritance, how traits are passed on,
Right.
and variability, how those traits can change. It looks at two core aspects that seem opposite, but are linked. Inheritance is about the faithful, exact transmission of genetic info from parent to offspring. It explains the constancy. We see why bacteria produce more bacteria like themselves.
Okay.
But alongside that, there's variability, change,
Yeah.
expressed in the offspring. These changes relate both to shifts in the actual genetic makeup and to environmental influences. It's this interplay between copying faithfully and occasional changes, mutations, that drives evolution.
So what's the difference between what an organism is right now and what its genes allow it to be, potentially?
That's a crucial distinction. Phenotype versus genotype. The phenotype is the expression of the genotype. It's the observable characteristics which you actually see or measure at a given moment. Think of it like the performance of a play. Phenotypic changes are often temporary, influenced by the environment. We sometimes call them physiological adaptations, like a bacterium changing its metabolism based on available food.
So phenotype is like changing your clothes based on the weather, but your underlying DNA stays the same.
Excellent analogy.
Yeah.
Exactly. In contrast, the genotype is the underlying genetic constitution of the cell. It's the heritable total potential characteristics determined by the DNA in its chromosome, the blueprint. The genotype stays constant during growth, barring mutations, and the functional unit within that genotype is the gene. Genotypic changes are actual heritable alterations to the blueprint itself, rewriting the script.
But that blueprint isn't totally fixed, is it? Sometimes typos happen, accidental changes. That's where mutations come in, providing the raw material for evolution.
That's right. A mutation is fundamentally a change in the nucleotide sequence of a gene. Think of it as a spelling error, A single letter changed, added, or deleted in the genetic instruction manual. A cell showing the effects of a mutation is called a mutant.
How do these changes happen at the molecular level?
They occur as changes in the sequence of those purine and pyrimidine bases in the DNA. We generally categorize them into two main types based on the kind of error.
Okay, the first type sounds pretty disruptive, changing the whole meaning.
That would be frame shift mutations. These happen when one or more nucleotides are either added insertion or lost deletion within a gene. They're so disruptive because the genetic code is read in three-letter words called codons. If you add or delete a letter, it shifts the entire reading frame from that point onwards.
So all the words after the error are wrong.
Exactly. All subsequent codons are misread, which almost always leads to a completely non-functional protein because the amino acid sequence gets totally scrambled.
Ouch. And the other type, point mutations, sound more subtle.
Point mutations are indeed more localized. They result from the substitution of just one nucleotide for another at a specific spot in the gene sequence, like changing just one letter in a word. But even that single change can sometimes have significant effects.
How so? How do these single changes affect the protein being made?
Well, what's fascinating is how these subtle nucleotide changes impact the translation process when the mRNA code is read to build the protein. There are three main outcomes from point mutations affecting translation.
Okay, what are they?
First, a missense mutation. Here, the altered gene triplet leads to an mRNA codon that specifies a different amino acid than the original. Depending on how critical that amino acid is, this might result in a faulty protein or maybe one with slightly altered function.
Okay.
Second, a nonsense mutation. This one's usually pretty bad. The altered triplet creates a stop codon prematurely in the mRNA. This tells the ribosome to stop building the protein too early, leading to a truncated, usually non-functional protein fragment.
Like hitting stop halfway through the instruction.
Precisely. And third, there's a neutral mutation. Here, the altered triplet results in an mRNA codon that, due to the redundancy of the genetic code, actually specifies the same amino acid as the original. So even though the DNA changed, the protein sequence doesn't. It's a silent mutation. The cell doesn't even notice the typo at the protein level.
So how do these mutations actually happen? Are they totally random errors, or can things cause them?
While some mutations do occur spontaneously, often as errors during DNA replication, many are actually induced or their frequency increased by external factors. Damage from things like UV light or X-rays, forms of radiation, can cause mutations. An agent that significantly increases the mutation rate is called a mutagen.
So you can induce mutations?
Yes. Mutations caused by mutagens are induced. We use mutagens in the lab to create mutants for study. A major effect of UV light, for instance, is causing pyrimidine dimers. It makes adjacent pyrimidine bases in the DNA strand, like thymine, stick together abnormally. This distorts the DNA and makes it hard to copy correctly, leading to errors or mutations.
Are there chemical mutagens too?
Absolutely. We group mutagenic chemicals based on how they mess with DNA. Some chemicals react directly with DNA, chemically altering its bases. Others are base analogs. They look so much like normal DNA bases that the cell mistakenly incorporates them during replication, but then they often pair up incorrectly later, causing a permanent mutation. And another type are intercalating agents. These are flat molecules that slide themselves between the DNA base pairs, like sticking a card into a deck. This distorts the helix shape and often causes those frame shift mutations, insertions, or deletions, leading to major protein errors.
Okay. Understanding this seems critical for things like antibiotic resistance or even cancer. How often do these mutations actually occur? What's the rate?
Exactly. Understanding mutation rates is vital. The mutation rate is the probability that a specific gene will mutate during one cell division, or the average number of mutations per cell, per division. It's usually a very small number, expressed as a negative exponent, like 10 to the minus something. For a single gene, it might range from 10^3 down to 10 to 9 per cell division. Tiny probability.
Can you give a real-world example? Like for antibiotic resistance, that's a huge problem.
Sure. This directly impacts things like resistance. For example, the mutation rate for penicillin resistance might be around 10 to 8 per cell division. That means roughly 1 in every 100 million divisions might produce a penicillin-resistant mutant.
Still sounds rare.
It is per division. But think about bacteria. They divide every 20 minutes sometimes, and there can be billions in an infection. Even at that low rate, resistance can emerge relatively quickly just by chance mutation. For streptomycin resistance, the rate's a bit higher, maybe 10 to 6, 1 in a million divisions. These numbers highlight why resistance is such a persistent challenge. Evolution in action, unfortunately, for us sometimes.
Okay, so mutations are random changes. But can bacteria actively share genetic information? Swap genes. This gets us to genetic recombination, right? A faster way to adapt.
It is indeed a major driver of rapid adaptation and evolution in bacteria. Genetic modification or recombination is about forming a new genotype by rearranging genes, usually through an exchange of genetic material between two homologous chromosomes carrying the same types of genes.
How do they exchange this material?
Bacteria have three main ways to transfer genes between cells, allowing them to share genetic innovations.
What are the three ways? How do they work?
Okay, first is transduction. This is when bacterial DNA gets transferred from one bacterium to another by a virus specifically, a bacteriophage acting as a delivery vehicle.
A virus taxi for DNA.
Pretty much, a tiny viral taxi. There are two types. Generalized transduction, where basically any piece of bacterial DNA can accidentally get packaged into a phage particle and delivered. And specialized transduction, which is more specific. Only bacterial genes located near where the phage DNA integrates into the host chromosome can be picked up and transferred.
Okay. Viruses as DNA couriers?
Oh,
What's the
okay.
second way? More direct contact.
Yes, the second is conjugation. This involves the direct joining of two bacterial cells, allowing for mating. Genetic material, often on a plasmid, gets transferred from a donor cell to a recipient cell through a specialized bridge, a pelus.
Bacterial sex, essentially.
You could call it that, yes. A classic experiment showed this with E. coli. They took two strains, each unable to make a specific nutrient, auxotrophs. When they mixed them, some offspring appeared that could make all nutrients, prototrophs, proving genetic material had been exchanged and recombined between the cells. Direct sharing.
And the third way, no contact, no virus.
The third is transformation. Here, a bacterium takes up naked DNA directly from its environment. This DNA might come from a dead, broken open bacterial cell nearby. The recipient cell just absorbs fragments of this free-floating DNA and can incorporate it into its own genome.
Like finding useful instructions lying around.
Exactly. Griffith's famous experiment with pneumonia bacteria demonstrated this harmless bacteria became deadly by picking up DNA from dead pathogenic ones.
These natural processes sound incredibly similar to what scientists do in the lab with genetic engineering, right? Harnessing these mechanisms.
Precisely. These natural gene transfer mechanisms are the foundation for genetic engineering. That field involves deliberately changing an organism's genetic structure through biochemical manipulation, often called recombinant DNA technology. It uses tools like plasmids and phages as vectors to cut and paste genes, introducing new genetic material into cells to give them new capabilities like making bacteria produce human insulin or creating disease-resistant crops. It's applied microbiology built on understanding these fundamental natural processes.
Okay, bacteria are a huge part of microbiology, but just one piece. Let's shift focus to some other fascinating microscopic players, starting with fungi. We know mushrooms, but the microscopic fungi, molds, and yeasts are everywhere too.
They absolutely are. Fungi are a vast, incredibly diverse group of eukaryotic organisms. Their cells have a true nucleus, unlike bacteria. They're of great practical and scientific interest.
What do they look like microscopically?
Well, their appearance is incredibly varied. You see them as that velvety blue-green stuff on moldy oranges, or the white fuzzy patches on old cheese or jam, or the furry growth on stale bread. And of course, the larger mushrooms. Morphologically, they're very diverse. They are eukaryotic, spore-bearing. They crucially lack chlorophyll, so no photosynthesis, and they reproduce both sexually and asexually.
Right, no chlorophyll, so they don't make their own food like plants or algae. What's their main role then, especially the saprophytes? Why are they so important ecologically?
Their importance is immense. Fungi are heterotrophic. They need organic compounds for nutrition. Many feed on dead organic matter. These are the saprophytes, and this is where their critical ecological role comes in. Saprophytes are nature's primary decomposers.
The recyclers again?
Exactly. They break down complex dead plant and animal material into simpler substances, returning essential nutrients to the soil, increasing its fertility. Without fungal decomposition, ecosystems would grind to a halt, buried in dead stuff. Beyond that, saprophytic fungi, especially yeasts, are vital in industrial fermentations, making bread, beer, wine, cheese. And while some fungi do cause diseases in humans and animals, these are generally less common than bacterial or viral infections, though some can be quite serious.
You mentioned some are dimorphic. What does that mean? How does it help them?
Dimorphism is a fascinating adaptation. It means the fungus can exist in two distinct forms, usually depending on the environment or if it's in a host. For example, it might grow as a single-celled yeast form under some conditions, like in lab culture or maybe in the bloodstream. but under other conditions, perhaps inside host tissues, it grows as a multicellular filamentous mycelial form, a network of thread-like hyphae.
Like having two modes.
Exactly, like the fungus Taphrina. You can switch between these forms. This ability can be crucial for survival, dispersal, or causing disease. The fungal body, or thallus, can be a single yeast cell, which has a true cell wall for structure, or it can be that complex mycelium.
How do they reproduce? Molds seem to pop up everywhere. Mushrooms appear overnight.
They have very effective reproduction, using both sexual and asexual strategies. Sexual reproduction starts with connecting two compatible cells and fusing their protoplasts. There are various ways this happens. Gametic copulation, fusion of sex cells, gametangel copulation, gametes from different structures, spermatization, transfer of a male nucleus, somatic copulation, fusion of regular body cells, gametangel copulation, fusion of specialized reproductive organs.
Complex. And they produce spores from this.
Yes, sexual reproduction leads to various types of sexual spores like ascospores, oospores, zygospores, basidiospores. These often allow for genetic recombination, mixing genes up for better adaptation.
And asexual reproduction, that seems like how mold spreads so fast on my leftovers.
Asexual reproduction, also called somatic or vegetative, is indeed very efficient for rapid spreading. It doesn't involve fusion of nuclei or sex cells. It's basically cloning. Methods include fission, cell splitting in two, budding, a small outgrowth becomes a new cell like yeast, fragmentation, pieces of the fungal hyphae break off and grow, and general spore formation.
Asexual spores, too.
Oh yes, tons of them. Various types like candidiospores, sporangiospores, chlamydospores, oidea, blastospores. These asexual spores are often produced in huge numbers and are easily dispersed by air or water, which is exactly why mold can colonize new food sources so quickly.
Okay, from fungi, let's move to algae. Often just seen as pond scum or seaweed, but they're way more important, right? Especially for oxygen.
Immensely important and often vastly underappreciated. Algae are a diverse group, mostly unicellular or simple multicellular organisms. The key thing is they contain chlorophyll, like plants, and perform photosynthesis, turning sunlight into energy.
How are they different from plants, then?
Mainly in their reproductive structures. Algae have much simpler structures for sexual reproduction compared to complex plants. They live pretty much anywhere there's water and light. Seas, oceans, lakes, ponds, streams, even damp soil, rocks, tree bark. Some are even endophytic, living inside plants symbiotically.
And their role?
Their photosynthetic activity is vital. They are the primary producers in most aquatic ecosystems, forming the base of the food web. They convert CO2 into organic matter and release massive amounts of oxygen, a huge contribution to the air we breathe.
What do they actually look like morphologically? Are they all just green slime?
Not at all. Many are single cells, and these can be club-shaped, spherical, rod-shaped, spindle-shaped, lots of variety. Others are multicellular, forming filaments, sheets, colonies, every conceivable shape and complexity. Think of the variety in seaweeds alone.
And their cells, like bacteria or fungi.
Most algal cells are eukaryotic. They usually have a rigid cell wall, though often thin. But there are cool exceptions. Diatoms, a major group, have cell walls made of silica, basically glass. Makes them thick, rigid, and often incredibly intricate, like tiny jewel boxes.
Wow, glass houses.
Pretty much. And some motile algae, like euglena, don't have a rigid wall, but a flexible outer layer called a periplast, letting them change shape. We classify algae based on things like their pigments, which cause their different colors, red, brown, green, their photosynthetic storage products, the type and arrangement of their flagella if they have them, their cell wall chemistry, and their reproductive structures and life cycles.
How do these vital producers reproduce, these oxygen factories?
They use both asexual and sexual methods. Asexual reproduction is often simple vegetative cell division cloning, very efficient for rapid growth when conditions are good. Aquatic algae often produce unicellular spores for asexual reproduction, which can disperse easily.
And sexually, do they fuse cells?
Yes, sexual reproduction involves the fusion of sex cells, gametes. This blends genetic material, creating diversity before a new generation forms. The fusion product is called a zygote.
Are the gametes always the same?
Not always. If the fusing gametes look identical, we call it isogamous. If they're different in size or shape, typically a large non-motile female egg and a small motile male sperm, then it's heterogamous, similar to reproduction in more complex organisms.
Okay. Next group, protozoa. The name means first animals, right? Suggests they're movers and eaters.
It absolutely does. From Greek protos, first, and zoon, animal. A good description for these diverse eukaryotic protists. They're generally single-celled, Key traits are their ability to move actively at some points in their life, and crucially, they lack a cell wall.
No cell wall. Makes them flexible.
Yes, gives them flexibility. While mostly microscopic, some form colonies, but these are just bundles of independent cells, not truly multicellular. The study of them is protozoology. They're hugely important ecologically as grazers on bacteria, controlling microbial populations. Unfortunately, some are also major parasites, causing diseases like malaria, giardiasis, sleeping sickness, affecting millions worldwide.
What are their cells like inside, simple or complex?
Quite complex, actually, much like other eukaryotic cells. Their cytoplasm is separated from the environment by a cell envelope, and they have one or more nuclei containing their DNA. The cytoplasm itself is often differentiated into two zones.
Zones.
Yes, the inner endoplasm, which is more fluid and contains most organelles, and the outer ectoplasm, which is clearer, more gel-like, and often involved in movement, like forming pseudopods in amoebas or feeding. This organization allows for complex functions within that single cell.
And how do these first animals reproduce?
They typically use both asexual and sexual methods. Asexual reproduction is usually simple cell division. Binary fission means splitting into two daughter cells. Multiple fission means one parent produces many daughters, allowing rapid increase.
And sexually?
Various types of fusion of gametes occur in different groups. A common process is conjugation, where two individuals temporarily join and exchange nuclear material, like swapping genetic info before separating and dividing. You see this in paramecium, for example. It boosts genetic diversity.
Okay, last category. The really enigmatic ones, bacteriophages and viruses. On the edge of life itself, you said, powerful agents of infection. Let's start with bacteriophages, the bacteria eaters.
Yes, phages. They have a fascinating discovery story. Frederick Tort in England, 1915, noticed bacterial colonies sometimes just disappeared, and he found this effect was transmissible. Then Felix Durrell in Paris, 1917, independently saw the same thing, this autolytic effect, and he coined the name bacteriophages, literally bacteria eater, great name. Their discovery revolutionized our understanding of microbial interactions.
What do they look like? Are they like complex cells?
No, much simpler. Incredibly minimalist but effective design. All phages have a nucleic acid core, either DNA or RNA, never both containing their genetic instructions. This core is covered by a protective protein coat called the capsid.
Just protein and nucleic acid?
Fundamentally, yes. The capsid isn't one piece. It's built from smaller protein subunits called capsomeres. And capsomeres are made of even smaller protein bits called protomers. It's a self-assembling modular design.
Do all phages look the same, like that classic lunar lander shape?
That's the most famous thing. But no, there's remarkable diversity. We group them into about 6 main morphological types based on their structure. That complex one you mentioned has a hexagonal head, a tail with a contractile sheath for injecting DNA, and tail fibers for attaching.
Okay.
Others also have hexagonal heads but might lack the contractile sheath, or have very short tails, or no tail at all. Some are just simple filaments. This structural variety often reflects how they infect their specific bacterial hosts.
And the broader category, viruses in general, they cause so many human diseases.
Yes, viruses infect everything. Humans, animals, plants, even bacteria and archaea. They're responsible for illnesses ranging from the common flu and chickenpox to devastating diseases like smallpox, polio, rabies, HIV/AIDS. Some cancers are virally linked. They even impact bacterial infections like tuberculosis, their effect on health, agriculture, ecology. It's immense.
What's the basic structure of an infectious virus particle?
A virion? A virion, the infectious particle, generally have 3 main potential parts. First, the nucleic acid core, DNA or RNA, holding all the genetic info for making more viruses. Second, the protein coat, the capsid, protecting the nucleic acid.
Okay.
And third, some viruses have an outer lipid membrane, the envelope, which they usually steal from the host cell membrane as they exit. But importantly, most viruses don't have this envelope. They are naked viruses, relying just on their capsid.
So if they're just nucleic acid and protein, maybe with a lipid coat, what do they actually do? How do they cause disease if they can't reproduce on their own?
That's the key point. They can't reproduce on their own. Viruses are obligate intracellular parasites. Meaning they absolutely must get inside a living host cell to replicate. They lack the machinery for making energy or building proteins themselves. They are completely dependent on the host cell's biochemical machinery.
So they hijack the cell.
Exactly. Their main function is to get their genome, their genetic code, into a suitable host cell. Once inside, the viral genome takes over. It redirects the host cell's machinery, its ribosomes, enzymes, energy, to start producing viral components. More viral nucleic acid, more viral proteins. These components then self-assemble into new variants.
And this damages the cell.
Often, yes. This parasitic replication process exploits and frequently destroys the host cell, releasing the new viruses to infect more cells. That cellular damage and death is what causes the symptoms of viral disease. It's a microscopic, hostile takeover, repeated millions or billions of times.
Wow. So what does this all mean? We've taken quite a journey through this unseen world, from tiny bacterial shapes and their crazy metabolisms to the complexities to genetics, mutations, gene swapping, and these viral hijackers. We've touched on fungi recycling everything, algae making oxygen, protozoa moving around.
We really have covered a lot of ground.
It seems the big takeaway is the sheer diversity, the essential roles, good and bad, and the incredible complexity packed into these microscopic life forms.
Absolutely. And if we connect this to the bigger picture, understanding microbiology isn't just science stuff. It's about appreciating the fundamental, often hidden processes that govern all life on Earth.
Right.
We've seen the vast diversity, the critical roles, the mind-boggling complexity within things too small to see. This deep dive, presented by Knowledge Flow, really shows how these basics are anything but simple. They're the foundation the whole living world is built on.
Yeah.
So this raises a question for you listening. How does knowing these details about the hidden lives all around it and inside you change how you think about your own body, or your food, or even the air you're breathing right now, knowing it's full of these tiny architects?
Definitely food for thought. And maybe one last thing to ponder, given their incredible adaptability, how fast they reproduce, how they thrive in extreme places, what previously unimaginable role might these microscopic organisms play in shaping the future of our planet, and maybe even humanity itself, in ways we probably can't even conceive of yet?
It's a captivating thought, isn't it? The potential is as vast and unseen as the microbial world itself.
Absolutely. Well, thank you for joining us on this deep dive into the invisible universe of microbes. Until next time, keep exploring the wonders of the world, both the seen and the unseen.